major focus in my lab is bringing in systems biology to tissue engineering to try to study disease and understand it and develop better approaches to therapeutics. My view of systems biology became much deeper when I myself got involved accidentally in this very interesting systems biology project. And I came into it thinking it would be a tissue engineering project, but to really make progress, we had to do some systems biology first. So what is the project? So I work with a gynecology surgeon, and he's very passionate about the stagnation that has occurred in the field of endometriosis research. Endometriosis is a disease where little bits of the uterine lining grow outside the uterus in the abdominal cavity, and it can be invasive, actually very much like cancer. And it causes tremendous pain, infertility, and a lot of suffering, and it's really underdiagnosed. The symptoms often occur early in teenage years, but it takes about 10 years for the average patient in the United States to get diagnosed. You can only diagnose surgically. And so Keith Isaacson, who's at Harvard Medical School and Newton Wellesley Hospital, and my collaborator, approached me and he said, you know, we really need better models of this disease. We want to model how this, so it turns out that every woman during her monthly cycle, bits of the uterine lining actually go in the abdominal cavity. And so we know that this happens in every woman, so why do 10% of them have that tissue show up and grow there and live there and invade? Very mysterious question still. So Keith Isaacson said, well, can we tissue engineer a model of the peritoneal lining and put some of these cells on there and see how this invasion happens? So we actually got a nice foundation grant for that, very grateful to a foundation that wants to remain anonymous. And we started working on it, and what we realized is we knew very little about what's happening in the fluid in the peritoneal cavity. When you look at the literature, people had studied inflammation and so on, but they hadn't done it in as systematic a way as you would if you were involved in systems biology. So a collaboration between Doug Lothenberger, who's one of the founders of the field of systems biology, Keith Isaacson, the surgeon, and myself, we set out to do a clinical study and really do a comprehensive investigation of the inflammatory environment in the peritoneal cavity. So there's immune cells there's these endometrial cells, and there's all these molecules. And so what we did is we said, well, gee, the really important thing is the way these cells are communicating. So let's look at the cytokines that they're using in a really multiplex way. So not 3 or 4 or 5, let's look at 50 of them and see if we can learn about the networks of inflammation in these patients. Moreover, let's think about it. You know, what surgeons do is they classify patients stage 1, stage 2, stage 3, stage 4 based on what the disease looks like during surgery. Are there a lot of lesions? Are they big, little? And yet, so when we started thinking about this, the first approach was, well, we'll see if there are cytokines that associate with particular stages of the disease. That must be what it is. But then we took a step back and we said, let's learn from our experience in cancer systems biology, where 2 patients with tumors that look very similar could have really different disease, and you would use different therapies. So if you're HER2 positive, you take Herceptin, and if you're not, you don't, and you may have a different therapy. If you're like me, triple negative, you would do other things. So what hadn't happened in endometriosis is thinking about it in an unbiased way where you separate the surgical appearance from the molecular. And there have been people who've started to do this, but we saw an opportunity to do this in a really comprehensive way with the immune networks. So what we did is we took a whole set of patients who had all stages of the disease, and then the control patients, and we measured 50 different cytokines. And then we put all the patients together and we used a multivariate analysis. And so instead of just looking at individual things going up and down, we said, are there things that move up and down together in an individual patient? We did this, we actually found a very interesting thing. And this is actually very exciting finding, and I've been invited all over the world to talk about this, and surgeons are very excited about it. Because what happened is we had 2 groups of patients, and one had a signature of 13 cytokines that changed up and down together, okay? Now that's very important because it means that they're probably part of a network. Moreover, we took that information on those cytokines and we went to a huge database that's a genome database, a gene expression database, where many different Genentech and a lot of foundations and government agencies have published the cytokines that different immune cells secrete. So there's already that database. We can use that database, don't have to measure it, and we can say what immune cell network is consistent with the cytokines we found. So we used that huge database and reverse engineered the network and found that macrophages in a particular state were probably the culprit. We then went in and took samples from patients, put them in culture. Lo and behold, they were making the cytokines, and very importantly, we found that we could inhibit their production by inhibiting a pathway that no one had really linked before to this process. And so now we've gone all the way from this sort of naive first step, thinking we're going to do a tissue engineering project, to really saying, in order to understand the process, we have to know what's going on in a network way. And having a model of that interface is important, but unless we understand the cues that these cells are getting, we don't have a prayer of recapitulating the tissue. This approach to studying the process of endometriosis, maybe the origins and progression, is so new. We submitted it to Science Translational Medicine because it is something important for people to start thinking about. Here's a whole new way to classify the disease, not to usurp what the surgeons do, but to complement and add to it. So this work is viewed in the field now as a breakthrough work and was published in Science Translational Medicine, and already we have 2 collaborations to follow on, because a big question is, we have one patient population. There could be differences in the disease process and other patient populations that we couldn't tease out in our studies. So we have a big study going on in Brazil right now with Mauricio Brau to study his patient population, and with the Infertility Center in Norway to study their patient population. We may find similar kinds of processes or different ones. The point is, we've galvanized a whole international community to think about this disease in a new way. I presented this work at the American Association for Gynecologic Laparoscopy, which is the main society for all the laparoscopic surgeons, in November of 2013. And after I gave the talk, after the session was over, I had people mob me and ask me to come give talks on this all over the world, because this really does represent, to people who've been in the field for their whole careers, a new direction. And it was a fantastic experience because you really do feel like you have the chance to make a difference. So I've gotten invited to Moscow, to Vienna, all these really good places. Moscow, they wanted me to come in January, but that was a no. So I've gotten invited, and I'm giving a keynote. I got invited to give a keynote lecture at the American Society for Reproductive Medicine that'll be held in the fall of 2014 on the basis of people who've been in the field for a very long time really seeing the value in this now that they've had a chance to look at it and really think about the approach, and particularly the ramifications for pooling patient populations and really teasing out some of these networks with a systems biology approach.